Rahim. This is lecture number 4 about a series of lecture about protein metabolism. In this lecture we will discuss formation of ammonia and its metabolism, how it is formed. Now, what is transaminase or aminotransferase? And what is transamination? Transaminase is an enzyme which transfer amino group from one molecule to another, like ALT alanine transferase, which is also known as GPT, glutamate pyruvate transaminase. It catalyzes the transamination of the transamination of amino group from alanine to alpha ketoglutarate to form pyruvate plus glutamate, while AST aspartate transferase are also known as GUT glutamate oxaloacetate transferase. It transfer amino group from aspartate to alpha ketoglutarate, and this can be shown in this reaction as L-alanine plus alpha ketoglutarate in the presence of PLP as a coenzyme. ALT transfers amino group from alanine to alpha ketoglutarate to form pyruvate plus glutamate. In the second reaction, in the presence of PLP, AST transfer amino group from aspartate to alpha ketoglutarate to form oxaloacetate plus L-glutamate. So this is very important reaction for the removal of ammonia from any amino acid. Now, what is the significance of this reaction? As we can see, in both these reactions, a common molecule is formed, a common amino acid formed. This is called L-glutamate. What is the significance of L-glutamate? That it can easily undergo oxidative deamination. So this is very important. Now, significance of transaminase. So in the liver we can easily deaminate this L-glutamate. Now, significance of transaminase is that in the liver and heart disease their level is increased, which is helpful in the diagnosis of liver and heart diseases. Now, what is deamination reactions? As we have already discussed about transamination, now how this amino group is removed from this L-glutamate and other amino acids. In the liver, amino groups from different amino acids are collected in the form of L-glutamate molecules, as we have already discussed. In hepatocyte, L-glutamate is transported, where it undergoes oxidative deamination with the help of L-glutamate dehydrogenase, which require NADP or NADP. L-glutamate dehydrogenase is an enzyme located in the liver hepatocytes. It oxidatively deaminates the amino acid L-glutamate, and the specific property of this enzyme is that it can use NADP as well as NAD. It is the only enzyme in our body. Now, what thing control oxidative deamination and how this reaction occur? This oxidative deamination is controlled by level of ADP, ATP, NADPH, and NADP as well as NAD. If there is increased level of NADPH, its activity will decrease, while if there is low level of NADPH and higher level of NAD or NADP, its activity will be increased, while in the presence of ATP it is inhibited, while ADP activates it. Means high energy inhibits it and low energy in our body activates this enzyme. Now, why this reaction is of significant importance? Because this reaction deaminates. It provides energy. It provides amino acid for energy. And how it is compartmentalized? How this reaction is compartmentalized? The enzyme L-glutamate dehydrogenase is located only in the mitochondria, and the toxic ammonia is and this reaction is compartmentalized, means only occur in the mitochondria. So we can eliminate, we can prevent the toxic effects of ammonia. So this reaction only occur in the mitochondria, and ammonia cannot damage the tissues. So this is also an intoxication reaction. Now, how this reaction occur? This reaction can be shown in this. It is not some clear, but L-glutamate in the presence of NAD or NADP, L-glutamate dehydrogenase deaminates L-glutamate to alpha-ketoglutarate plus N-amine plus ammonium group. Now we will discuss about some other things on the following page. Why glutamate dehydrogenase level is increased in plasma? किस तरह how its level is increased? Its level is increased in different types of disorders. Now, amino acid oxidases and how they play their role. How amino now, as we have discussed about this reaction, this is one reaction that oxidative deamination occurs. In this, there was removal of hydrogen group in the form of NADPH and NADH. Now another oxidative deamination, and this is by amino acid oxidases. For example, there is alpha amino acid, and this amino acid oxidase act on this alpha amino acid in the presence of R2 oxidizable FMN flavin mononucleotide, and in this also oxidation occur. A removal of hydrogen group is oxidation, so FMN is converted into FMNH2, and their alpha amino acid is formed, which is unstable intermediate. It adds water and is converted into alpha K2 acid plus NH4 plus ammonium group, while FMN H2 combines with O2 to form H2O2, which is the help of peroxisomal catalase converted into water plus oxygen atom. Now, non-oxidative deamination, how it occurs? It is specific for serine and threonine, not for all other amino acids. And why serine and threonine can be non-oxidatively deaminated? It has OH group on alpha carbon, so this specifies them for this reaction. Serine dehydratase in the presence of PLP convert it to pyruvate, which is alpha K2 acid plus NH4, and threonine dehydratase in the presence of PLP convert it to alpha K2 glutarate plus NH4. So this reaction, this is. In the next lecture we will discuss about some other thing. This reaction can be shown there, that is this reaction is very important. This reaction FMNH2, this is a amino acid oxidase reaction, and this is non-oxidative deamination by amino acid. That how this occurs? Serine dehydratase in the presence of PLP convert it to pyruvate plus NH4, while threonine in the presence of threonine dehydratase is converted to alpha Q2 butyrate plus NH4. And clinical significance of glutamate dehydrogenase was remaining. GLT is present in normal serum in trace amounts, but increased activities are observed in case of liver disease in which hepatocellular damage is present. So in the case of hepatocellular damage, its level can be increased in our plasma. So this is also a diagnostic material against the hepatocyte damage. Now this is the reaction that how this enzyme act. Actually, this is the reaction. Glutamate in the presence of NAD or NADP, NAD or NADP uh, have removes in oxygen hydrogen, and in this way this is oxidative reaction because NAD is reduced to NADH and our NADP is reduced to NADPH, while L-glutamate is oxidatively deaminated. So this was very important. Subscribe our YouTube channel for more such type of important